Let's jump right in. Have you ever actually stopped to think about how we get pure, sweet, white sugar crystals out of giant, messy green sugarcane stalks? Or, on a totally different scale, how a doctor can spot something as complex as malaria hiding inside a single, tiny drop of blood? You know, these are everyday mysteries, but they both point to one massive question. How do we pull the invisible answers out of complex, tangled messes?
Welcome to This Explainer. I'm super excited to dive into this with you because the secret to solving these everyday puzzles lies in the fascinating and often invisible science of separating mixtures. Today, we're going to look at exactly how science untangles chaotic blends into pure, usable, and even life-saving substances.
Here's our roadmap for today. We'll start with the magic of mixtures, then move on to sorting liquids with science, followed by colors and centrifuges, and finally, how we're purifying our world.
All right, Section 1, the magic of mixtures. Let's set the stage.
So, looking at the contrast here, it's pretty stark, right? On one hand, you've got a homogeneous mixture. Think sugar completely dissolved in water. It's totally clear, perfectly uniform throughout. Every single sip is going to taste exactly the same. But on the flip side, you have a heterogeneous mixture, like muddy water. It's chaotic, it's not uniform, and you can literally see the particles just waiting for gravity to drag them down to the bottom.
Let's expand on this a bit. Homogeneous mixtures are everywhere. Vinegar, your favorite aerated soda, and get this, even solid metals can form homogeneous mixtures when they're melted down together. We call those alloys. And once they cool, good luck trying to separate them with just physical methods. On the other hand, heterogeneous mixtures are your classic sand and water or oil and water. Oh, and don't forget gases. If you've ever walked through dense fog or thick smoke, you were literally walking through a heterogeneous mixture suspended right there in the air.
Moving right along to Section 2, sorting liquids with science.
Okay, so here's our first real powerhouse tool, the distillation apparatus. It looks like a mad scientist's dream, but we use this specific setup for a very particular problem. Basically, when two liquids mix together completely, meaning they're miscible, but, and this is crucial, they have boiling points that are at least 25 degrees Celsius apart.
Let's break down the mechanics here. First, you crank up the heat on the flask holding your mixture. Because of that 25-degree difference we just talked about, the liquid with the lower boiling point vaporizes first. It literally leaves the other one in the dust. Then those hot vapors travel through a long glass tube called a condenser. This uses circulating water or air to cool them right back down. Finally, the cooled vapors condense back into a pure liquid, dripping safely into a completely separate vessel. It is just such an elegant, foolproof solution.
But you know, what happens if the liquids just flat out refuse to mix? If they're immiscible, like mustard oil and water, we actually get to skip the heat altogether. Instead, we use a separating funnel. Since oil and water have completely different densities, they naturally form distinct layers. The lighter, yellow mustard oil happily floats on top, while the heavier water sinks to the bottom. To separate them, you literally just open the stopcock and let gravity drain that bottom layer right out. Easy as that.
Up next is Section 3, colors and centrifuges. Let's look at some of the more advanced techniques.
First up, paper chromatography. I love this one because it honestly looks like magic, but it's pure science. You use a liquid solvent to carry a substance up a strip of filter paper. Because different components interact differently with the paper, it turns into a kind of microscopic race where every runner has a different top speed. This is exactly how chemists untangle the hidden individual colors trapped inside green spinach leaves or even just standard black ink. Now, what if you're dealing with particles in a liquid that are incredibly fine and just stubbornly refuse to settle? Enter centrifugation. Picture a playground merry-go-round. If you spin around really fast, you feel a massive outward pull, right? Centrifugation taps into that exact same centrifugal force. You spin a mixture in a tube at mind-boggling speeds to create a massive outward push, which separates the components entirely by their weight. When the machine revs up and those tubes swing horizontal, that extreme outward force shoves all the heavier particles all the way out, packing them super tightly at the very bottom of the tube. Meanwhile, the lighter liquid just safely floats on top. To put that into perspective, this is exactly the process doctors use to separate light plasma from heavier red blood cells to detect diseases. It's absolutely crucial for modern medicine.
Which brings us to Section 4, purifying our world. Let's talk real-world applications.
Imagine you're trying to purify muddy river water. Sometimes those dirt particles are so incredibly fine that they take forever to settle on their own. But by adding a chemical coagulant like powdered alum, we essentially force those tiny, stubborn specks to stick together. We call this coagulation. Once they clump up into larger, heavier masses, gravity finally takes over. They drop to the bottom as sediment, and bam, we can easily filter off the clean water sitting on top.
Oh, and we definitely can't forget about sublimation. This is a really cool chemistry trick where a solid transitions directly into a vapor, literally skipping the liquid phase entirely. Say you have a messy mix of sand and a sublimatable substance like dry ice or naphthalene. You just hit it with some heat, the naphthalene instantly turns into a gas and vanishes into the air or a collection tube, leaving the plain sand behind. No messy liquids to deal with, it's a remarkably clean way to separate specific solid mixtures.
So, pulling our whole scientific toolbox together, look at how perfectly these techniques map to real-world problems. Muddy water? We use coagulation. Blood plasma? That needs the high-speed spin of centrifugation. Oil and water? Separating funnels, thanks to gravity. Hidden flower pigments? Chromatography reveals them. And a dry mix like sand and naphthalene? That's easily solved by sublimation. It's like a giant puzzle where every messy mixture has a perfectly engineered solution.
Now, you might be asking, why does all this actually matter? Well, the big takeaway here is that these aren't just isolated lab experiments. They are literally the foundation of our modern existence. As we speak, your own kidneys are using separation principles to continuously filter waste out of your blood. On a citywide scale, infrastructure relies on sedimentation and coagulation to treat millions of gallons of sewage before it's released. We use these exact principles to sort dry recyclable plastics from wet compost. And researchers, they're using advanced separation techniques to mine valuable lithium out of our old laptop and phone batteries. It is literally how we keep our world clean, running, and sustainable.
All of this leaves us with a massive challenge to ponder. As human beings produce ever more complex electronics and entirely new synthetic materials, we are constantly creating bizarre new mixtures that nature has literally never seen before. So what undiscovered separation techniques are we going to need to invent to clean our oceans and save our planet tomorrow? The science of separation is far from over. In fact, I'd argue it's more important right now than ever before. Thank you for joining me on this explainer. I hope this helps you see the invisible science untangling the world all around you. Stay curious and keep exploring.